Firstly, Aaron Puri, who got his PhD in chemical and systems biology at Stanford University, and where he worked on chemical tools to dissect host pathogen interactions involving enteric bacteria. He then did postdoc research at the University of Washington, where he was co-mentored by Mary Lidstrom, Pete Greenberg, and worked on genetic tools and chemical signaling in methane oxidizing bacteria. He's been an assistant professor at the University of Utah and Department of Chemistry and the Henry Eyring Center for Cell and Genome Science since 2019. And today's going to talk about quorum sensing signal diversity. So really looking forward to talk. Aaron.
Thank you, Nigel, for that introduction, and thank you, Nigel and Trent, for the invitation to be here. This is a great opportunity, and it's nice to hear all of the wonderful talks that have gone on so far. So as Nigel mentioned, I'm an assistant professor at the University of Utah, and what I'm going to talk to you about today is a project that's really been supported by the JGI, primarily through various community science programs for a very long time. So I'm going to try to emphasize that. And the main meat of what I'm talking about in the story I'm going to tell you today is through this JGI Functional Genomics Community Science Program Award, where I am working with two wonderful co-PIs. So Dale Pelletier is here from Oak Ridge National Labs, and Amy Schaefer, who's a senior scientist at the University of Washington. The three of us have teamed up to work on cataloging chemical signaling, and specifically quorum sensing signals in carbon cycling microbial communities here. So the overall question that we were interested in here, and I think that Trent and some others really set this up quite well, is this idea of, can we take a metatranscriptome, a metagenome from an environmental sample and predict the ways that bacteria are interacting with each other in the environment in that sample, right? So, the answer is we're working on it, right? And so one of the big missing links here is, of course, metabolomics, and that's something that's really cool about this functional genomics program. And so really at its core, the part of this that I'm interested in, especially coming from a chemistry department, is how do you predict which natural products are small molecules, specialized metabolites, secondary metabolites, whatever you'd like to call them. How do you predict which ones are produced from sequence, right? So for our test case, we're going to look at quorum sensing. Many of you are probably familiar with this, so I just want to make sure that we're all on the same page here and give you all a quick refresher. So this is the way that many bacteria can coordinate group behaviors in the environment with a relatively simple system. So in this system here, we have a signal synthase that produces the second component, which is a quorum sensing signal. In most cases, for the bacteria we're talking about, these signals can diffuse in and out of cells freely. And will sometimes interact with the third and final component, which are receptors, the signal receptors. So at low cell density, we have a low concentration of this quorum sensing signal. But then at high cell density, we have a much higher concentration and therefore more receptor occupancy. And these receptors are also transcription factors that will undergo a conformational change and then sit down on particular genome targets and transcriptionally control various programs responsible for group behavior. So the canonical one that many people have heard of is biofilm formation, right? But this can be a lot of processes usually where bacteria are trying to affect their environment. It wouldn't make much sense energetically for them to devote a lot of energy without this being a coordinated effort amongst multiple cells here. So for the bacteria we're going to be focusing on, which are gram-negative proteobacteria, the quorum sensing signals are these molecules called acyl homoserine lactones. So I'm showing you some example structures here. In these structures, you can see they all have this common homoserine lactone moiety. I promise I'm not that slow. But I think you get the idea here, the right side. And it's actually the left side of these molecules that confers the signal specificity. What I mean by that is which transcription factors will bind them, right? So these are sort of like languages. Different bacteria may use different signals to coordinate with kin. And the most common group of them I'm showing on the left there, and we give these names based on their chain length and the substituent at the third carbon here. So you can have a ketone there, a hydroxyl, or a methylene, a straight chain. And so you'd have something like this top one is just C8 homoserine lactone or 3-oxo C6 homoserine lactone. So to date, about 30 structures have been identified, starting about 40 years ago. And what's interesting is that there's very low amino acid identity between synthases. And so what that means is that it can be, even though this is a relatively simple system, it can be very difficult to predict which molecule is being made from a synthase, right? So this is like a test bed here, a very simple test bed for how can we actually predict what secondary metabolite is made from, using sequence. So a little bit more about the biosynthesis of these molecules. So that common homoserine lactone core, that comes from methionine via S-adenosylmethionine, but unlike, many of you are probably familiar with S-adenosylmethionine as a methylating agent, here we're actually using the backbone, which I think is kind of cool. So the 4 carbons from the backbone of methionine and S-adenosylmethionine get incorporated into this homoserine lactone core. And the donor of the chain, so the left part of this molecule, can be either an acyl carrier protein from fatty acid biosynthesis or a CoA thioester. And I'll get more into that towards the end of the talk. So whether or not that left side comes from fatty acid biosynthesis and an acyl carrier protein or a CoA thioester influences the structural diversity of the molecule. So we have, sort of what I would maybe think of as very formulaic structures on the left here. They're going to vary, again, in the length of their chain and the substituent, that 3rd carbon. We can get more structurally diverse molecules when we use a CoA thioester instead. And this is a relatively new discovery in the last 20 years or so, and I think is really exciting for expanding the structural diversity and therefore richness of the languages that microbes are using to interact using this system. So back to our overall question here about prediction of natural product production and therefore bacterial interactions from sequencing information. Myself and our co-PIs, we were very interested in applying this to things that we know and care about deeply, right? So we targeted 2 carbon cycling microbial communities that, again, have been supported by the JGI and various community science programs for quite a while. So the first one, which is not the one that I work on, but again, one of my co-PIs, Dale Pelletier, is here along with Mitch in the audience. They, at Oak Ridge National Labs, have been working with Populus deltoides, also known as the Eastern cottonwood, for quite a while. And this is a bioenergy crop, right, for biomass production. And there's a tremendous amount of resources that are available for this, which is really interesting, both for the host itself as well as the microbe side of the plant microbe interface. And this is part of this SFA from the Department of Energy about plant microbe interfaces. And this has been a very fruitful and long-standing research area. And I want to point out that there's actually a postdoc position available working on this project on the PMI. So please see Dale or Mitch or someone else later if you're interested in this. I think that would be a great team to work for. So there's a tremendous amount, again, of resources available here. So 561 isolates with sequenced bacterial genomes, 125 gigabase pairs of metagenomic sequence, all these things that were built up through these JGI community science programs that we could leverage for answering questions that we're interested in. So as Nigel mentioned, I did my postdoc with Mary Lidstrom and Pete Greenberg at the University of Washington. And the Lidstrom laboratory has been interested in methane oxidizing bacteria for over 40 years. And they have this great system, both Mary and Mila Shistosardova, which I think has been a long-standing JGI user. They've developed this system here where basically they take sediment from Lake Washington, which is in Seattle, and you can add it to a sealed tube and provide methane as the only carbon and energy source and create an enrichment you can passage over time a little bit here. And what's really interesting is they and others have discovered the fact that when you do this and you analyze the community that's formed, it's not just methane oxidizers, even though methane is the only carbon and energy source, but you get cross-feeding, right? So this has been used for studying these types of cross-feeding interactions for quite a while. And what's even more interesting to me is that when different researchers do this around the world with different samples, you get very phylogenetically similar cross-feeders. So there's some kind of association there that we're only now starting to understand the molecular mechanisms of. But again, this has been supported by the JGI for quite a while, a little bit more modest in terms of the numbers than the tremendous resources available for the plant microbe interface initiative, but there's also quite a bit available here. So we wanted to leverage these 2 carbon cycling microbial communities, so very much on scope for Department of Energy and JGI here, to try to look at this question. Can we predict which acyl-homoserine lactone quorum sensing signal is being made from the amino acid sequence of the synthase that makes it, right? So that's the relatively humble question here. And so to start out with, we wanted some kind of curated database. So Amy Schaefer at the University of Washington, again, one of my co-PIs, she took advantage of some of the downtime early during the pandemic when we had no idea what was going on. And in the evenings would kind of go through some of the literature and curate which signals had been reported as being produced by an organism where we knew what synthase was making the signal and what amino acid sequence was coming from the synthase. And I'll tell you, there are actually hundreds of reports like this, but many of them are using things like thin layer chromatography and bioreporter strains, which I'm not going to talk about here, but where, especially as a chemist, you're not quite sure if that's really what's being made. And I have an example up towards the end of the talk of even when we have quite a bit of information, how it can be difficult sometimes to assign the structure. But she did this work, which was fantastic, where basically she was looking for this link between sequence and product, and where we could have high confidence in the product. So basically mass spectrometry or NMR characterization here. And then she was able to use that information to figure out what kind of a amino acid cutoff we would need to confidently predict what signal was being made by a synthase given its amino acid sequence. So what I'm showing you here are some examples of sequence similarity networks, which some of you may be familiar with, but just to walk you through these in case you're not. In each of these clusters, you'll see a bunch of rectangles, most of which are blue, but a few of which are red, or pink, I guess. And each of those rectangles is going to represent an amino acid sequence. Now, if two amino acid sequences are sufficiently similar to one another, and we can choose what that threshold is, you draw an edge between those two. So what's wonderful is because Amy had put together this data set where we had high confidence in about 100 of these, she could go through and systematically check a bunch of different thresholds so that we could feel very confident in the threshold that we set here, which, if any of you care, is 85 alignment score threshold, unitless. But what we're looking at here is cases where by setting that threshold, we are able to see that our defined standards, which are being shown here in pink, they're clustering properly. So these are what we call isofunctional groups now, where we would feel confident that everything in each cluster is producing the same thing. So this would be the same as if you were working with any other kind of enzyme. I really liked how Patrick kind of expanded this to discussing just enzymes and enzyme function in general. But so again, just to repeat this so we're all on the same page, what we're looking at here are two metagenomes from, again, the carbon cycling community number 1, the populus microbe interface, as well as this methane oxidizing bacterial community on the right. And then by using this alignment score threshold, we can then predict in some cases what a bunch of different sequences are making, right? A bunch of different synthases. So for example, you might be interested in this particular blue one. We'd say that probably makes isovaluryl homoserine lactone because there are two characterized representatives in the cluster. So I hope that makes sense here. But what you probably notice and what spawned us to write this community science program proposal was there's a whole lot of clusters here in these communities where, again, I said we've done a lot of work on them where we have no idea what's being made. We can't predict. And I found this quite interesting because, again, relatively simple idea here looking at quorum sensing signaling. And quorum sensing signals have been known about and characterized in one way or another for 40 years now, right? So it's amazing to try to, or to see that even for something like this that people think we know all about, we have no idea, right? And can't predict these things. So we were lucky to get to work with the JGI to try to tackle this, and both with Yasuo's team in synthetic biology and DNA sequencing, and also Trent's in metabolomics. And we requested that they take 183 of these synthase genes that, again, I'm sorry to keep going back and forth with the delay, but most of which were from these undefined clusters, or clusters where there's no undefined or no defined representative. And we asked Yasuo and his team to synthesize 183 of these. And I believe this is one of the larger projects, not in terms, each of the synthases, its DNA sequence is only about 600 to 700 base pairs, but it's a lot of different constructs, right? I know for both Yasuo and Trent, it was a different type of project, and I appreciate them doing this. So these again represent both PMI and Lake Washington strains or from their metagenomes, and it's very cool that we live in the future and you just need the metagenome sequence, right? We don't need the isolate to be able to get these things. I think that's amazing. So I'm showing here a wonderful summer student who visited us from Skidmore College named Abigail Monahan, who took on the task of, after we received the 183 constructs, each of which contained a synthase, she went ahead, these are all in E. coli, and heterologously expressed each of these and extracted the supernatant, the exometabolome there, so that we could get those analyzed with Trent's team. So that is 183 times 2, 366 different samples. And here is Abigail very proudly and rightfully so standing next to her 366 extracts, which luckily were not lost by FedEx and made it back here. That was very terrifying. So we sent those back along with 24 standards, again, for Trent and his team's non-polar untargeted metabolomics pipeline, and Catherine Liu did a fantastic job working with us there. So I'm really lucky to go after Ming because he explains tandem mass spectrometry, and actually I'm going to talk a little bit in a second about using the mass query language that he's developed. But so, how are we actually going to take the resulting data set and identify acylhomoserine lactone signals in there so we can link them with the sequence that we heterologously expressed, right? That's the whole goal of this. So we're very lucky that acylhomoserine lactones have predictable tandem mass spectrometry fragmentation patterns. So you can see here for an acylhomoserine lactone, there are these 4 main fragments that we see, although I'm not going to get into it here. I'm happy to talk to people about it later. There are classes that do not fragment like this, so there's a lot more to be done. Keep me in business for a little while longer here. But so what we did is we used Ming's fantastic language, which even I can understand, to try to query our data to find these acylhomoserine lactones, right? So I'm showing you here an example for us that Ben Bowen and Trent's team here at Lawrence Berkeley Lab, as well as Joint Genome Institute, as well as Ming, helped us write. And so this we wrote after looking through the fragmentation patterns of the 24 standards that we had run. And so really what this is saying is, like, query all of the data here where all of the fragments have a 102 Dalton fragment that is at least 30% as intense as the highest one, and at least 2 of the 3 other fragments here, right? So we use that to find what was in our representative data set, and then search that throughout the whole data set, and made sure that the resulting features, when possible, matched the retention times and high resolution parent masses of the standards here. And so we did a bunch of quality control checks, and I want to point out, this is Ethan here. I'm a relatively new professor, and the best part of my job is getting to work with people like Ethan. He's fantastic. So he started working with us as an undergrad, has stayed on for an additional year, and taught himself with Ben's help R and Python to do all of this. So he's really awesome. It's fun to work with people like that. And so Ethan is able to provide a list of what each synthase might be making using the data set we got. So here I'm just going to show you a summary of what this looks like with one of Ethan's figures. And very gratifyingly, this worked, right? So I was very happy about this. So this is, I know on the right-hand side, it's a little bit small here, but what I'm showing you is a heat map. And these are basically the different types of the kind of quote unquote boring signals that you would see. So we have all the straight chain ones here from C4 to C18, and then the hydroxys, and then the oxos as well at the bottom there. And then on the top, we just have all of the synthases that are arranged by their amino acid sequence similarity. So I want to point out 2 different things. So the first is that if you look across some of these rows, you can see that very divergent sequences in terms of amino acid sequences of synthases can produce the same signal, right? So these are things, maybe it's convergent evolution, not sure, but we definitely would not be able to predict what those are, right? And the other thing is, if you look across any or down any column, you can see that when you heterologously express one of the synthases, you oftentimes will see several different acylhomoserine lactones that are produced, right? And so I have a little bit of experience working with these from their native hosts. And so I want to point you to sort of, what do we think about this when we see that a heterologously expressed synthase might produce multiple signals? And to do that, I'm going to look at this synthase the gene for which is MBAI, and we know from working with the methane oxidizing bacterium that makes this, that this produces 3-hydroxy-C10 homoserine lactone. So when you're just growing your native host, this is what's called a precursor ion scan, looking for that 102 Dalton fragments. And what you can see is this mostly makes 3-hydroxy-C10 homoserine lactone, but the methane oxidizing bacterium will make a little bit of this 3-hydroxy-C12, right? So maybe 5%, 1 to 5%. When we heterologously express MBAI in E. coli, we get, about 70% 3-hydroxy-C10 homoserine lactone, and then it kind of goes down from there. But we also see C14 and 3-hydroxy-C8. So this is interesting, and I think that this is an ongoing question, especially because we have such great metabolomics data. Is like, what is the real answer here? What is the signal that's being made? And so it's nice when you have native producing results so that you can make a better call at that. But I think we can hopefully both agree, or we can talk later about that the primary signal being made is 3-hydroxy-C10 homoserine lactone, and we can start with that as sort of defining what would be made for a particular synthase. The other thing that is, of course, important is, can we actually trust these heterologous expression results? We're taking synthases, and they're codon optimized for heterologous expression in E. coli. We're taking these synthases from a lot of different, very diverse bacteria, heterologously expressing them in E. coli. Are they going to make the same things? This is very preliminary data. You can see that we need to work on the scaling here for intensity. But what I'm showing you is an extracted ion chromatogram for the mass of this 3-hydroxy-C10 homoserine lactone, actually made by a completely different bacterium where we could not predict based on amino acid sequence was being made. You can see the standard in blue, the heterologous expression in red. It makes quite a bit. And then hopefully you can trust me. Here we have a little tiny nubbin from the native producer, and we just need to work on doing this. And what we can do is we actually have 92 native producing strains of these 183. So we can go through and validate these, which I think is really cool. So the last part of this here that I want to try to draw some conclusions of is, are there actually some synthases which are more likely to produce their signals when heterologously expressed in E. coli? So I'm going to talk about this and then one more brief thing. So I told you that the left side of these molecules can come from an acyl carrier protein or from a CoA thioester. If they come from an acyl carrier protein and fatty acid biosynthesis, I might feel a little bit better about heterologously expressing your synthase in E. coli. E. coli make fatty acids. I'm sure the pool composition is different, but we might feel okay about that. But what about these CoA thioesters? Well, that poses a kind of an additional level of complexity here, because you need to usually have some sort of enzyme that's responsible for making that CoA thioester, usually from an exogenous carboxylic acid. This certainly isn't the only way that you can make a CoA thioester, but it's one of them. So what this means is there's another enzyme required besides the acyl homoserine lactone synthase to make your signal. And that additional enzyme is probably not present in E. coli. We don't really know, right? So we're taking one more step in our reductionist approach of trying to predict what natural product is made from. Now, maybe, Nigel, can I call this a biosynthetic gene cluster now? If it has two genes, I'm not sure, and they're not usually linked in the genome, but let's look at this in a little bit more detail, because the cool thing is we can actually predict from genome sequence, from KEGG orthology identifiers, in many cases, whether a synthase is using an acyl carrier protein as a donor or a CoA thioester here. So in this table, what I'm showing you is we actually ran, so again, 2 independent replicates for each of our heterologous expression strains. This is where we get 366 from our 183. And so this is actually a very stringent way of looking at the data that I can get into in more detail later. But if you look at cases where the 2 replicates match versus where they don't match, and you look at the synthases that are predicted to be linked to acyl carrier proteins, so again, that's the fatty acid biosynthesis more likely to, we would hypothesize, to be made in E. coli, 56% using this very stringent way of looking at the data of the ACP-linked synthesis gave clear results, but only 38% of the CoA-linked ones did. So I think that's maybe supporting our hypothesis here that it makes more sense to if you heterologously express these in E. coli, this is something you need to think about. And of course, it's really exciting to think about JGI and CRAGE and all these new heterologous expression hosts coming on, or maybe co-expressing CoA ligases with these synthases to try to fill this in a little bit more, right? So the last thing I want to say very briefly here is, what about finding new signals that people haven't seen before? So of the 94 synthases that gave clear results, so again, where the independent replicates matched, 22 of those don't match any of our standards for acyl homoserine lactones. And I want to show you an example here. So this is on single quadrupole, so it's nominal mass. It's a little bit dirtier, but this is again an extracted ion chromatogram for the mass of a compound that even at high resolution would match 3-oxo-C12 homoserine lactone. But so we thought, again, this was something that we could annotate. It's from this methylotroph, which I love methylotrophs, and from our data set here. So we just wanted to do something very similar to what I showed before, where we thought they would all line up, and that would be great. And this is a very common class of synthases that we're working on also for a very different project. We were very surprised when the heterologous expression and our native producer, it didn't match in retention time to the standard. So this makes us think that, again, since the high resolution mass matched, this is a constitutional isomer. So same atoms arranged in a different way here. And it also highlights the importance of having LC data, right? In addition to your standards. So I'm very lucky to get to work with a lot of very motivated grad students, and they were excited about the idea of finding a new signal here, right? So we grew up about 30 liters of this methylobacterium strain, and then did a bunch of 2D NMR to solve the structure de novo of this signal, which again, we had also been interested in because it seems to be made by a synthase that's very widespread in these methylobacteria. And so here it is. Here it's this 3-hydroxy 5Z, so it has a unsaturation here, C14 1 homoserine lactone. And so this is interesting because, again, we're basically populating the data now with more signals that can be predicted from synthase using these functional genomic platforms, which I think is really exciting. So just to conclude, we're trying to take steps towards being able to predict from metagenome, metatranscriptome, isolate genome sequences, what secondary metabolites are being made, and for me, most importantly, how we can draw inferences about how they're using those molecules to interact with each other in their environment, right? And the cool thing is that DNA synthesis enables us to do this when we don't have access to the strains, right? And I'm really excited because we have the potential to have doubled the number of synthases that are linked to their products with high confidence here. So again, there were 98, and we have 94 new ones with clear results. Again, I think it actually could be even more. And then at least now the heterologous expression host is important, and we know that, and that's something that we can investigate in the future. We can also use data sets like this as a toehold to start to discover new signals. So what's next? Again, we have all these native strains. And then I would love to talk to people here about what do we use this data set that we're trying to grow that's relatively modest in size, but maybe now it's 200 high confidence links of the signal product with the synthase, which again, even someone who's been working on quorum sensing for a while, I would have thought it would have been a lot larger with me starting my postdoc, but it's not. But is that big enough to maybe start predicting what would be made from sequence here? And then I think that as important, if not more important, I would love to make it so that when a synthase is flagged in a genome, it also says, the highest, this has X% identity to this one, which we have a confident annotated signal for, right? So again, I think this is a wonderful meeting to get to talk to people about that. So thank you very much for your attention, and I'd be happy to answer any questions. Again, I want to thank my co-PIs and other wonderful collaborators.
Great, thanks, Aaron. We've got time for one quick question.
Thank you for a great talk. I do have a quick question. Sorry. I'm curious about the diversity that you talked about, and maybe a bit beyond your talk here, but do you think there's a biological reason for that diversity, given the goal of, like, when you think of biofilm formation and quorum sensing, that it doesn't necessarily matter who's in your biofilm, maybe, and so that diversity of signal maybe is helpful? I'm not sure.
Yeah, I think that's a wonderful question. And also a step back, what constitutes a quorum, right? With this idea that Steve Lindau and others have done some wonderful work with. I think that you can classify, we heard a wonderful talk this morning about classifying different relationships between types of microbes as antagonistic or helpful. And I think that you can see both types of relationships among spectral strains in communities. And maybe classifying all of those for the diversity of signals you're talking about would be a great way to get an answer to your question. I don't have one. I think that the first approximation is they're not always cooperative at like the level of beyond kin, let's say.